Hey everyone, welcome back. In the last video, we installed Nextflow and Conda, and today I want to introduce you to a collection of best practice pipelines you can run with these tools, a community project called NFCore. Let's get started. So what is NFCore? NFCore is a global community of scientists, engineers, and students building open source, standardized bioinformatic pipelines that anyone can use. And there are currently over 100 pipelines covering many types of analyses, from genomics to proteomics to metagenomics. So let's take a look at how to explore them. So I'm on the NFCore website. You can be too by pointing your browser to nf-co.re. And to browse the available pipelines, we can click on the View Pipelines box in the middle of the page. And from here, you'll see a large collection, over 130 pipelines. So let's go ahead and sort them by the most popular to see which pipelines the community is using the most. So you'll notice pipelines for bulk RNA-seq, variant calling from whole genome sequencing data, single-cell analysis for 10x genomics libraries, and metagenomic assembly, and many, many other pipelines. And each card here links to detailed documentation about the pipeline. So let's open one to explore how it's structured. Let's go ahead and click on the RNA-seq pipeline. And what I really like about the NFCore documentation is that every pipeline has the same general layout, which makes it easy to learn about each pipeline because once you've learned about one, you know what to look for in the next one. And a good place to start is on the introduction page. Here you'll find an overview of what the pipeline does, the type of data it accepts, and what it produces. For example, the NFCore RNA-seq pipeline takes FASTQ or BAM files as input, performs quality control, trimming, sequence alignment, and outputs a gene expression matrix for downstream analysis. You'll also find a workflow diagram showing each step of the pipeline and which tools are used. And I think this is a great way to get a 30,000-foot view of what the pipeline does and how it works. And at the bottom of the page, you can find a credits section to see who developed the pipeline and how to cite it in your publications. Next is the usage page, and this explains how to run the pipeline. And in my opinion, the most important thing to look for when running an NFCore pipeline is the sample sheet. And the sample sheet is essentially a small table listing your samples, file paths, and experimental design details. And sample sheets are going to look a little bit different for each pipeline, as you can see here. So definitely make sure to take a look at this section of the documentation before running anything. Another important section is called the core Nextflow arguments section, and this controls how the pipeline runs. And one of these arguments is called profile, and it tells Nextflow how we want to manage software dependencies and environments. So if we want to do that using Conda, for example, then we can specify that here. If we want to use something else like Docker or Singularity, we can specify that here too. There's another handy argument called resume, and this is really nice because it allows you to pick up a run where it left off if for some reason something failed. In other words, you don't need to start over. The next page is the parameters page, and this lists all available options for customizing the workflow. From input and output options, to reference genome options, and tool-specific options for retrimming, filtering, processing UMIs, alignment options. There are even options for skipping parts of the workflow that you don't want to run, which can save both time and computation. So before running any NFCore pipeline, it's worth reviewing which parameters are required, which are optional, and what the defaults are. Next up is the output page, which shows what files each step of the pipeline produces. So for example, if we look at the FastQC tool, we can find a short description of the tool, what it does, and a drop-down describing each output file. And typically, FastQC results are stored in a folder called FastQC with 2 files per sample, an HTML report with quality metrics, and a zip archive containing tab-delimited quality control statistics and figures. So knowing what outputs to expect is important so that you can plan your downstream analysis. In earlier versions of NFCore, there used to be a results page with example outputs from test runs, and these were hosted on Amazon Web Services. So it looks like this feature was removed, likely due to cloud storage costs, or maybe it hasn't been updated yet, but that's okay. Finally, there is a releases page, which lists version updates and release notes. This is really useful for keeping track of new features, bugs, and what's changed between different pipeline versions. That's it for this video. I hope you've learned a little bit more about what NF Core is, what pipelines are available, and how the documentation is structured. In the next video, we're going to run the NF Core RNA-seq pipeline using Nextflow and Conda. I'll see you there.